Our first speaker will be Fabrizio Lombardi, who is currently a postdoc in the Patrick group in IST Austria. The group uses biophysics and information theory to understand the principles and mechanisms behind information processes in networks. And today, Fabrizio is going to present a talk about quantifying the coexistence of neural oscillations and avalanches. So, the floor is yours.
Thank you so much, and thank you for the invitation. So, as she said, the talk today is about the coexistence of two different phenomena in brain dynamics. I would say these two phenomena, neuronal oscillations and neuronal avalanches, are, I would say, antithetical, and I will explain why in a second. So on the one side, you have the neuronal avalanches, the neuronal oscillations, right? And whenever you think about neuronal oscillations, you think about something that has a characteristic time and also a characteristic amplitude. For instance, we have slow oscillations, the delta oscillations that you have during slow wave sleep, for instance. They are slow and they have high amplitude. Instead, if you take already faster oscillations like the theta or gamma, they are faster and they have smaller amplitude, right? So they have characteristic scales. On the other side, we have neuronal avalanches, which are cascades of neural activity that do not show any characteristic scales. They do not have any characteristic durations, and they do not have any characteristic sizes. Right? So, and for this reason, I will refer to them as a scale-free phenomenon. So, on one side, we have the scale-specific brain oscillations, and on the other side, we have the scale-free neuronal avalanches. Now, an interesting question is, what is the relationship between scale-free neuronal avalanches and scale-specific oscillations? And in particular, an interesting question is how the oscillations actually influence the dynamics of avalanches. Right? This is an interesting question, but I will not focus on this today. I'm sorry for that. Because what I want to do actually is to develop a minimal physics model that explains the coexistence of avalanches and oscillations, which is simple enough to be analytically tractable. And in that way, with this analytical tractability, we can connect the model to data and hopefully to have some testable prediction. I don't know if universal, but I mean, like, we hope to have some prediction for the model that we can test on data. So I will start and give you an introduction a bit on neuronal avalanches and brain oscillations, and in particular on related models. Right? And I will introduce our model, the adaptive Ising model. And I will compare the behavior of the adaptive Ising model with the MEG of the awake resting state in the human brain. And then if there is time, I will say something about the relationship between the dynamical relationship between oscillations and avalanches. But let me start from avalanches first. So I hope the video works. So here you see a recording from cortex culture with 64 microelectrodes, right? And each of the dots that you see blinking here, they represent peaks over threshold of from the local field potential of each electron. Right? So you see that the activity is transient. You have like some cascade, and then there is some quiescent period, and that when you take the raster plot, right? I think you are familiar with this picture. So you see that the activity looks highly synchronized. So a period with high activity, and then overall you have quiescence across the entire slice. Right? But if you actually zoom in, what you see that the activity that looks synchronized at this scale, it's only near synchronous. And you can go down more and more, right? Until you find what I call a fundamental unit of this process, right? And it's actually what we call neuronal avalanche. formal definition of an avalanche is a sequence of consecutive time pins, epsilon, with at least one event in which the event in this case will be a peak over threshold, which is preceded by an empty beam and terminated by one. So if you take this picture for good, right, so you can see the activity in this culture as a sequence of neuronal avalanches. All right. So typically, neuronal avalanches are characterized by their size and their duration. So the size is defined as the number of events in the avalanche, basically black dots that you see here. And the duration is simply given by the number of time beams that belong to the avalanche times epsilon, which is time beam, the size of the time beam. So now we can look, for instance, at the distribution of avalanche sizes. And when we look at the distribution of avalanche sizes, we realize that the distribution is a power law with an exponent that is about 1.5. Right? So when I was saying before that avalanches do not have characteristic scale, I was referring exactly to this property, that the distribution of sizes and also duration, they follow a power law. And power law is actually the first fingerprint of scaling bias, because you can change the scale of the problem, but whenever, even if you, if you coarse grain or you go to find the scale, you will always see the same behavior, right? Oh, okay, fine. And there is what's important here is that you have, of course, you cannot have sizes larger than system size, right? And the system size in this case is 64, and you have here exponential cutoff around 64 electrodes involved in that. Okay, now what is also even more interesting is that if you look at different systems, different scales, you see the same picture, right? If you look, for instance, at the activity in the spontaneous activity in cortex of awake monkeys, you see that the distribution of avalanche sizes follows the same behavior. And if you go up to the MEG of the human brain, again you observe the same behavior. Right? So it looks like that we have something that is universal, right? So, but please take this, what I say here, as what I call as universal exponent with at least a pinch of salt, because you cannot really. So I will say something more about universality, but it's not. But it's more safe to say that we have something that is common across scales and systems. Okay? And what it looks like that for the sizes, you have an exponent minus 1.5, and for the duration distribution, you have a power law with an exponent minus 2. Right? Now, why all this should be interesting? Because scale-free, so the power laws and universality are fingerprints of critical dynamics. So they usually emerge in systems that are close to a critical point or a second-order phase transition. So in that specific point, the system has very peculiar behaviors, right? So for instance, if you take a simple ferromagnet and you have local interactions, when you are close to the critical point, you actually have spontaneous collective behavior that are emerging because you are close to the critical point, right? So indeed, the observation of neuronal avalanches revived an old theory, the critical brain hypothesis, that was developed that was proposed in the 80s. That was the theory says that the brain should work around criticality because criticality is giving optimal computational capability. This was based on some models, right? And the necessary flexibility to adapt, but also long-range correlations which are needed to have collective behaviors, the collective behaviors that we actually observe in the brain. Okay?
Sorry.
Now, yeah.
Maybe you'll get to this, but that point about criticality, the crucial issue in physics is that the interactions are local, and so long-range interactions are.
Yeah, right.
That's not true in neural systems, right? A single neuron
Right, that's
influences
good.
many others over a large region.
Yeah.
And so these are very easy to get in neural systems.
Yeah, but you know that, like, so I was about to say that this hypothesis is really controversial, and we don't know. Actually, there are questions that are still very much open, and is this actually criticality or not?
Sure.
So, and one problem is for instance, that one, right? But also, but in that sense, I could say that most of the connections are local, right? So you have long-range connections, but they are not as many as the local ones. But of course, you're not like in the Ising model, which is like, for example.
Right. And so we would need different measures of
Yeah. Okay, so as you see immediately, so you have you have criticism about this hypothesis, but I don't want to actually go into this now. I mean, like, and especially this also this. I mean, does criticality? I mean, if the system is at criticality, does criticality have actually a function? Can we measure that? So far, we don't know, right? And also there's the question about avalanches and other types of criticality, for instance, statistical criticality. And then there's a big question in that, and I will go to this point now, whether criticality is tuned or self-organized. Right? And this is a question that most of the work, the modeling work, which is actually what I want to discuss now, was driven by, right? And of course, when the neuronal avalanches were discovered, there was a theory that was just ready for that, right? It was self-organized criticality, right? So in the paradigmatic model of self-organized criticality is the same type, right? So you have a pile of sands, right? You slowly add grains, and then when the pile reaches critical slope, you have avalanches, right? After the avalanches, like after the system has relaxed, you keep on adding grains, and you're going to have another avalanche, right? This looks awesome. I mean, like you have power laws in this system. You have power law distribution for the durations and power law distribution for the sizes, and you have scaling relationship, and everything is beautiful. But the exponents are not the one that you see in from neuronal avalanches. Because, for instance, for the size distribution, you have -1, and there's another problem. The avalanches in the sandpile model are not correlated, right? And when you look at the distribution of Poisson time, it's an exponential, and clearly doesn't fit the picture of the neuronal avalanches. But this was not a big, and then finally you have the time-space separation. But anyway, this was not a big issue because it was realized pretty soon that you can actually do something with the original sandpile model. You can add some plasticity, you can add some inhibition, frustration in this context, difficult frustration in this context. And if you do that, you can actually reproduce the distribution of neuronal avalanches with the right exponents, minus 1.5 and -2 for the sizes and -2 for the duration, and you can even get the temporal dynamics, right? Which is even more difficult than the distribution which I studied. Right? And at the same time, it was realized that you can actually take spiking neural networks. You can have some dynamical synapses. And you can actually robustly reproduce the dynamics of neuronal avalanches. So this is beautiful, except that you don't have oscillations. There's no way in this kind of models you get oscillations, especially if you are inspired by self-organized criticality. This is a cellular automaton, and you don't get oscillations. I mean, like lately, there's been some attempt to get something close to oscillation, but still it does not compare at all with data, right? But there's some hope when you go to spiking neural networks. Right? But before going to this point, I will show an example of a spiking neural network where you can get both oscillations and avalanches. So let me say something about brain oscillations. I mean, like, let me give you some examples. I mean, like, prominent example, like gamma oscillation in the hippocampus, frequency between 30 and 90 Hz, is detected, they are detected using local field potentials. They're short-lived, and they couple, for instance, with thin oscillations. Another very famous brain rhythm is the alpha. The alpha is actually relevant in this talk because we're going to work in the alpha band. And alpha the characteristic time of alpha is about 100 milliseconds. It's related to quiet wakefulness. You also have some alpha during sleep. There's a beautiful hypothesis about the functional role of alpha, which is pulse inhibitions. So pulse inhibition that says that somehow alpha oscillations are related to modulation of population spiking. And of course, there are studies showing that alpha correlates with perception and with attention. Of course, there are also other important brain rhythms like delta that you have during sleep, but this is just to say that oscillatory phenomena in the brain are widespread and they happen at different time scales and in different subsystems. But unlike avalanches, they are scale specific. Okay. Now, if we go to models, there are many options, and in these models, the models that I will briefly introduce, they work very well when it comes to reproduce the specific features of, I don't know, in this case, for instance, the inhibitory circuit, it's very good to get fast oscillations, right? This was already mentioned before, the excitatory inhibitory circuit with feedback, right? And this is good for gamma oscillations, but also used to get alpha oscillations. And then there is the typical, you can use a collection of oscillators, coupled oscillators, and then when you use coupled oscillators, you can explore actually the spatial structure. You can have different network topologies. You have beautiful examples of synchronization, explosive synchronization, and things like that. Right? Now, the focus in this case, while in the model, when modeling neuronal avalanches, the focus was mainly about the dynamical regime, where you actually see the power law emerging. So in this case, the focus is mostly on biophysical constraints and control parameters of that drive and the control parameters to get these different oscillations in different subsystems and in different conditions, right? But even though the focus is more physiological, right, oriented, also in this context, the question of what dynamical regime actually, in what dynamical regime you can get fast or slow oscillation, or in what dynamical regime you can get actually both, depending on how you drive the network, it was always present, right? So now the question is, how can we bring together these two different worlds, right? The world of avalanches and the world of oscillations with the different modeling projects, right? So the first attempt was done about 10 years ago. I have to say, quite nice, right? And this model, the spiking model of excitatory and inhibitory neurons, typical structure. So you have an excitatory population, and there are local interactions. Then you have an inhibitory population, the local interaction. They interact. There are feedback between the inhibitory and the excitatory, and vice versa. So in this specific case, you have the inputs are integrated at the level of the synapses, and then there's a probabilistic trial. So there is some stochasticity in system. Right? So you can define avalanches in this model. You take the network activity as the total number of neurons that are spiking each time. So you have sort of random walk if you want. Right? So you take a threshold, right? And when the activity is above threshold, you say that you have an average, right? And you define the size as the area underneath the curve, and for duration you take, what is the zero crossing time for this run. So now what they show in this paper is that as a function of the inhibitory commutativity, that is the percentage of connections that inhibitory neurons have, and the excitatory commutativity, there is a sweet spot, or a sweet line, right, where you can actually get avalanches that show a distribution of sizes with exponent minus 1.5. And at the same time, you can get oscillations in network activity. And then if you take the envelope of this oscillation, so if you filter in the alpha band, you take the envelope, you can compare the model to image data, and qualitatively you have something similar, right? And so what they did, they measured the correlations of the envelope, right? And they show that the correlation can actually be described by some power law with similar exponents when you are in this cross. I mean, when you are in this. I mean, over this line, right? But that's the level of complexity, right? So the model is not analytically tractable. You cannot make. I mean, you have to rely on this phenomenon, like qualitative connection between data and model. It's not analytically tractable. They claim criticality, but it's ex post. I mean, the model has not critical point, so you cannot show anything. You cannot show whether it's a critical point or it's not critical point. You cannot talk about a dynamical regime in which you have this coexistence of avalanches and oscillations. Right? So what we want to do here is exactly to get something that is analytically tractable, because we want to say something also about the coexistence regime. So our model in reality will be in between something that is fully stochastic, as the Ising model, and something that is fully deterministic. So we're gonna, as a matter of fact, we're gonna use an Ising model, which is stochastic, and we're gonna add deterministic feedback to the Ising model. So I guess all of you are familiar with the Ising model, so collection of spins, but in this case do not we used to model active and inactive neurons. So we take the traditional Ising model. The traditional Ising model has no external field. I mean, you can also have an external field, but in the traditional, like equilibrium Ising model, there's no external field. So we take the Ising model. We start with zero external field. So then we have the total magnetization, we simply define as the sum of the spins divided by n, the number of spins. Right? And at the time step t plus 1 of the simulation, the magnetization becomes the feedback, becomes the external field of the Ising model. Okay? So now this field is not constant in time, but changes from one time step to another time step. And what is important is there is a minus sign. So if a spin wants to go up, flip up, so the external field tends to push the spin down. So the dynamics is the usual global dynamics for the Ising model. In this case, we have an external drive, which is given by the feedback. So if you want, you can think about the feedback as either as spike threshold adaptation or the effect of some inhibitory population over the excitatory population. And indeed, I mean, it can be shown that formally, to something when you get to the fully connected and the linear limit of this model, formally, you can map this model to exactly to what I was showing before. So two different populations, one excitatory, one inhibitory, with feedback, with cross population interactions. Okay, so now what we want to do, we want to go to that limit that makes the model tractable. And this limit is the fully connected limit and in continuous time. Because in this limit, you can represent the model as a system of Langevin equations. Right? Still, I mean, the second equation is the one you have seen before, and is the one for the feedback. First equation is non-linear here, right? So we have to do another approximation, so we have to go to the linear approximation in order to solve this system analytically, right? So now, we linearize the equations around the stationary point m equals 0, h equals 0. So in the limit c that goes to 0, of course, you get the standard Millfield Ising model, which is a critical point at beta equal 1 for g equal 1. So, with feedback instead, you have this second order equation, which is a van der Pol oscillator. And van der Pol oscillator has an antiphon of bifurcation at beta equals 1. So now the feedback, so you go from the Ising model is an equilibrium system. So the feedback that is changing in time is mapping an equilibrium problem transition to a dynamical transition to oscillations. As I said before, I mean, like, this is fully equivalent to having an EI system. Now, having the analytical solution, we can draw the phase diagram. So we can solve that equation, we can get the eigenvalues, and we can get the phase diagram. Right? So now, the interesting part of phase diagram is for beta lower than 1, the critical point. Because for beta above the critical point, you simply have oscillations, self-oscillations. Right? So nothing really interesting. But here you have something interesting because so if you are like below this red line, which is c equal beta minus 1 divided by 4 beta, right? So if you have two negative real eigenvalues, you have exponential relaxation, you have a constant movement process, and you see that autocorrelation of the magnetization simply indicates exponential. Right? Now, when you are in this regime instead, you have two imaginary eigenvalues. You are in the resonant regime, and you start to see intermittent oscillations. And these intermittent oscillations become more and more prominent as you approach the critical point. Right? So if you look now at the autocorrelation function of the magnetization, you have something that you have a cosine that is, you have an exponential that is modulated by cosine. It's a clear sign that you have oscillations. And then when you cross the critical point again, you just have an oscillator. So we are interested in characterizing the activity around the critical point. So here is some simulation, right? 2D simulation. You see that this is pretty much random. You start to see some structure here. And then this is at the critical point you start to see pretty much oscillation, and that is a full oscillatory regime. Right? Then there is another regime when you cross a certain value of beta, which we did not explore, which called relaxation oscillation. Basically, you have that abruptly the system is either going up or down. Right? And here I'm showing you how the magnetization and the drive and the field go together. Right? So basically, you have the drive, which is the field, and it's equal to the magnetization at time t minus 1. Okay, so what we want to do now first is to characterize what we call the network activity near the critical point, and see if there is something interesting. To do that, I mean, we use the usual quantity that you use to characterize the random walks. Basically, the zero crossing time and the area under the curve. We plot the distribution of this quantity. You see that when you're close to the critical point, right, so you have power law in both cases, in both quantities, both for the area and for the zero crossing time. Right? And when you move away from the critical point, so you lose that dA. I say, of course, I mean, what I'm showing here also, this is for very weak feedback. But if you increase the strength of feedback, you start to see that oscillations somehow become prominent, right? So the scaling behavior shrinks, and you see a bump on the tail of the distribution. This is especially evident here for the zero crossing time. Right? And also, I mean, like the power law behavior is relatively robust for small values, I mean, at least for 2 orders of magnitudes of C. But then if C becomes too strong, I mean, like you have deviation on the scaling chain. Okay, so now what is important is like we want to connect, as I said, we want to connect the model with the data. And now having the analytical solution, you can actually do that in a way that is not qualitative, but it's just like you can actually connect them. Like we don't need to just say, okay, this parameter, I mean, like if we use this parameter, I mean, data looks a bit better, closer to data, we can actually infer the parameters from the data. So we have imagery recordings, 4 minutes, 14 subjects, 273 millimeters. So on the model side, we can calculate the autocorrelation function. The autocorrelation function is in terms of gamma and omega. Gamma and omega are expressed as a function of beta and C. Right? So what we can do, we can fit the model correlation function to the correlation to the autocorrelation of the simple sensor activity. Right? And from this fit, we get beta and C, right? As you see here. So the fit is quite good, I would say. And we get a pretty narrow distribution of beta and C values. So beta is is like around 0.99, very close to 0.99, slightly subcritical. Oh, all right. Slightly subcritical, and C is very close to 0.01. So we're gonna also use for the simulation 0.99 and 0.0. Now we look at the dynamics at single sense level. First, oscillations that you see at single sense level in the alpha band, as they did in the EI model I showed you before. Right? This DMG, this is our model. This is still qualitative. By eye, they look pretty much similar. But then we try to quantify this. And we do what we have done before. So we take a threshold, so we define the zero crossing area. Right? And we take the distribution of log, what it was shown before. So why log sizes? I mean, we decided to go for log sizes because when you have like sizes, when you have a quantity that stretches over many orders of magnitudes, that's the way to go, and that's the way it is done in granular materials, for instance. That was our reason, but like maybe there are other reasons to do that, and you get it. I write here wiggle, but it's actually, it's close to a gamma, right? And please notice. So the green is like 14 subjects, right? And the purple is more. Independently of the threshold, because you have data collapse when you use different thresholds. Right? So we are pretty close. There's a kind of shift. We don't really match here, because of course, the data are more complex, there are no linearities and other things, but we are pretty close, a little bit close. And also interesting, so beta in our model measures how close to the critical point you are. There is an independent measure of criticality, which is the scaling of the correlation function. It's alpha. So the closer you are to 1 with alpha, the closer you are to the critical. And you see that there is pretty much there is a very good correlation between beta and alpha. These two things are independent. Right? And it's very good correlation. Okay, so I said more or less everything. This correlation, another interesting thing, it looks like that criticality goes with alpha power. So it looks like that, as it was suggested in the past, I mean, like, alpha oscillations are somehow related to closeness to critical. Okay? Now, last thing that I have to show you is that we also get neuronal avalanches. Right? But before, since I mean, he was talking about gamma, I'm showing you an intermediate step to get to neuronal avalanches, which are extreme events. Right? So we parse the system in subsystems to mimic the structure you have in magnetic currents. Like we have 100 subsystems. In magnetic currents you have 50 more. This is not really crucial, actually. So again, you have extreme events. Extreme events are calculated as peaks of the threshold. We use exactly the same technique, exactly the same parameters, same threshold, same bin. Everything is the same, both in the model and in the field. Right? So we have raster plot, you can group them, and like, so you can define the activity per bin, right? And you can define also the quiescent time. I'm running, sorry. This is the quiescent time, and the activity per bin is simply defined as the number of extreme events that you have in time bin. So, in green you see the energy, in purple you see the model. They are independent of bin size. There is a data collapse when you rescale everything by the average activity of the network. Right? It's a pretty close match. We fully match with error bars, both for the activity, for the probability of activity, and especially for the probability of inactivity. This is extremely difficult to match. Not just because we did it, but you can check other models. I mean, this is something no one looks at because it's not a disappointment. Right? Anyway, so I skip like details here. I just want to show that this is just for scaling relationship, nobody cares. And so you know already the definition of neuronal avalanches, and I just want to show you that we go pretty close also to the distribution of avalanche sizes, and we also go pretty close to the distribution to the scaling relationship between durations and avalanche sizes. Okay, so you see that if you're not close to the critical point, you don't match the distribution of our avalanche sizes. All right, so I'm done. So we have tractable moment. So we get both avalanches and oscillations, but of course there are many questions. Effects of connectivity, spatial structure in jail, right? Inhibition, how to link this to spiking activity. So can we go from the mesoscopic picture to the microscopic picture? In principle we can, because we have control on the spins, on single mirrors, but we haven't done it. And those, I mean, it would be very nice to see what is the link between our model and previous model for the existence of avalanches and oscillations. Thank you. This is a big link. And this is collaboration with Selva, Oren, Daniele, and Gabriel. Thank you so much.
Thank you, Fabrizio. Any questions? Arne?
Yeah, so regarding this exponent, so I didn't get a very good description for why you have matching exponents in the network models, but not in the sandpile model, for instance. So I found the sandpile model very, very intriguing because it's a very simple model that you can easily grasp. So could you somehow modify the sandpile model in order to get closer to the exponents? So
Yeah.
for instance, by changing the friction of the grains, or
yeah, we are.
I better understand what's actually going on in the process.
Right, so we actually have done that. You can do that. Right? So you can, so basically what you need is to allow some dissipation because in the original sandpile model, so you have the grains and transfer to the connected nodes of the network, like, and it's not dissipation, right? So one thing that you have to add is like some dissipation, right? And also the other thing that you may think of is inhibition, right? So because in sandpile there is no inhibition, right? But if you talk in terms of like neuronal networks, you may think that there are cases in which you're not transferring, like somehow you're lowering even more the energy of the site that is next to the site that is relaxing energy. Right? So you can, you can do some. We have done that, but then you have to. So you have to do it in ways that you have inhibition and plasticity, which is like modulating somehow how much energy you transfer from one side to the other, right? Which is somehow, so you can think of it as dissipation, right? Because if you don't transfer, because of plasticity, you don't transfer 100% of the energy. You sometimes you transfer 50, and sometimes 75 or 25 or nothing, right? That's pretty much close to the idea of having plasticity, right? Something that, so you have to add these two ingredients. Nice.
So we have time for one very quick question, if anyone has any. So in this case, we can thank Fabrizio again. So our next speaker today, the last speaker of the first day of our workshop is Cristina Savin, who is an assistant professor of neuroscience and data science at NYU. And her group is using machine learning and statistics to figure out how learning and memory happen at the level of neural circuits. She's using different models such as Gaussian process, maximum entropy, and online learning RNNs. And today, Cristina will talk about making sense of neural responses during naturalistic behavior.
Jade, I'm sorry for that. Still the same thing. Thank you, Arnolina, for inviting me. I had a really good time today. I enjoyed all of the talks. So it's good that we're ending on this talk because it kind of straddles topics in both of the sessions. So it's going to have elements of modeling pairwise correlations between neurons, but also focus. The second part of the talk is going to focus on data dynamic system models, both of which were specifically focusing on the naturalistic behavior kind of context and what extra complexities does that bring to the discussion. Okay, so before I start, I should acknowledge the people that work on this. So this is work done in a collaboration between Dora Angelati and Malla at NYU, spearheaded by two postdocs in our respective labs, Eduardo Balzani, who is the of the fancy machine learning, and Jean Paul Noël, who has collected data, and so it has been a core force in driving the interpretation of our results in neuroscience. Okay, so let's get started. And I put something funny, a quote here, just to echo Manisha's point early in this morning that no action that an animal takes, like each action that an animal takes is unique. So it's driven by complex and ever-changing circumstances, and traditional neuroscience has willfully chosen to ignore that fact by restricting variability in action and behavior as much as possible to drive the collection of something that is as close as we can to perfect repeats of the same situation that you can do statistics on easily. Now, this is not the only way of doing neuroscience, and there's a trend emerging in recent years, driven by technological advances to some degree, to stop doing that and try to embrace this neurovariability as a new and possibly richer window into understanding brain function. Okay, so I put this up as it's a picture from a review by Cristina Savin, my friends, where they're really arguing that we need to move past these sort of simple stereotypic behaviors into the realm of ethologically relevant behaviors, which is where the dynamic regime where the brain was optimized to actually behave. So we're starting to sort of look at the brain in the regime that is intended to. Okay, so even if you sort of like not go into the woods and start looking at animals doing their thing, there are a lot of technical advances that make the behavior that we're collecting more sophisticated or more complex than before. First of all, people are trying to design tasks that are at least more intuitive to the animal, where generalization is possible across variants of the same task. It's a review from Jean-Paul Noël that I really like, so a bit over there. But on the technical side, like we have VR setups that are increasing the complexity of the environment in which we embed our animals. We have rat farms and automated training of animals at scale that allow us to make rodents do things that 5-10 years ago we had thought that they can't do because they're just stupid. It's just like you train them long enough and you don't have to like, pay 100 students to do it. It works. We also in the private universe that has been sort of like very stereotypical in its way of doing things, people are starting to move away from the traditional paradigm and starting to doing freely moving monkey experiments. And here I put a picture, the monkeys are real, the room is of a big arena at NYU where we're recording activities wirelessly for monkeys as they're doing foreign new tasks. And it's not really the jungle just yet, but it's still a lot richer and a lot more complicated than what we would have done in the past. Lastly, with this sort of focus on, there's a lot more general focus on behavior. Like we're using a lot more cameras to characterize what the animals are doing, even outside the context of the stereotype task that we ask the animal to do. And this brings new machine learning tools that characterize these extra measurements in sort of complex behavioral feature spaces that we can use to understand what's going on under the hood. So the data is getting a lot more interesting, but there's a problem. It also gets like extremely complex and high-dimensional in ways that we never had to deal with before. So it becomes a really important practical question is, okay, we've collected all the data, what do we do with it? How do we make sense of responses given all this complexity? And you should notice that because there's no stress structure, there are no repeats, no two tries are similar enough to be averaged over, this means that all of the statistical tools in the experimentalist repertoire don't work anymore. You can't estimate tuning functions. It's very hard to even do a PCA. In what kind of space, what does that mean? So this is the statistical regime that we're operating in, and I'm going to present a couple of tools that we've developed trying to start addressing these problems. And they're not solutions. I think that they do their job to some degree, and you guys should be inspired to take over and make them better. Okay, so for all of the results that I'm going to present today, I'm going to use one particular data set collected in the Angelaki lab, which is not quite natural behavior, but rich enough to do the job, and we call it the firefly task. This is a nice picture of a firefly. This is more of a cartoon of how it looks in reality. So the monkey sits in a virtual reality screen kind of thing, and it controls the joystick. The task is something like catching a firefly. So transiently, the target comes on. I should stop pointing with the example line. So the target comes on, which represents the firefly showing up. It stays around for a bit and then it disappears. The monkey needs to control the joystick to go from where it was to the target. If it's close enough to the target, it going to get the reward. If it misses, it's not going to get the reward. And what it has to go on is the information in working memory about what the firefly was doing, and it has to integrate optic flow that comes from these little triangles that tells you sort of like path integration, tells you how fast it's moving in this virtual reality space. Okay. So again, very far from the jungle, but it has some interesting properties. First of all, the monkeys found this intuitive, and there are two examples why we know that this is the case. So first of all, motivation. Like a good teenager, they spent hours doing this. The juice is kind of secondary. They actually are bored enough to want to do this for fun. So that's good. The other thing that's interesting is that they vary, and this is the behavior. So they're doing almost perfectly. There's some biases. They tend to undershoot, just kind of a least effort principle. They need to get the reward. They don't need to get the target perfect. So they stop really. But they get the job done. What's the other thing that's interesting is that you can tweak the task. Like in this particular example, we're changing the density of the optic flow, so we're diminishing or impoverishing their sensory information that they're getting, and they can do it right away. But you can do other things. You can add more than one of the fireflies at the same time. You can do multiple variations of the same task, and the monkey, because it knows how to move already in virtual reality, will switch to new task constraints on the time scale of the session. So you don't need to train animals for weeks or months in order to switch the task. You can do it right away. So this is sort of like telling us that, okay, this is an interesting regime, and maybe capture something about what the brain does. The other thing that's interesting is this task combines elements of multiple brain computations that have been cited in isolation. So like working memory, sensory integration, path integration, sensory evidence integration, and also this computing of the latent variable where the firefly is. So it allows us to ask sort of pretty basic questions about how far did the traditional approach take us? Is the whole the sum of the parts? Are we seeing the same kind of phenomenology that the textbook tells us while looking at this slightly more complex task, where the computations happen in the context of other things going on at the same time? Okay? So we're particularly interested in this sort of manipulation of the sensory experience because it also allows us to ask more probabilistic questions about to which extent the brain relies on internal models and sort of how it weighs the sensory evidence as a function of its reliability, things like that. But ultimately, what we really care about is what does the brain do in the context of this behavior, where there are quite a few things going on at the same time. Okay, so the first thing that if you're experimenters that you want to know about your brain activity is tuning. And in this particular context, we would want to know individual neurons that we're measuring in the brain, what is it about the task that they care about, okay? So this is how the textbook version will tell you how to do it. But this is an example from the hippocampus by Castle and all, where they're recording from the hippocampus and they want to know which of the spatial variables the animal running around, head direction, etc., are driving individual neural responses. So how they're going to do this is they're going to fit models about all possible combinations. Does the neuron care only about position, head direction, etc. All of the combined models here. And then they're going to do model comparison to figure out which of them is actually good. So this tells you if this particular model is winning, then basically that means that this neuron is sensitive to this configuration of 3 possible task variables. Now, with only 4 task variables, it already gets quite annoying. You need to fit a lot of models. But in our case, we have 20 plus. So this is not going to work for very, very obvious reasons. So what do we do instead? So we put together this framework that is sort of a little Europe's paper published a couple of years back that tries to address specifically this issue. So first of all, what's a graphical model? So we're going to try to model spiking responses of single neurons using something that's almost like a GNN. So you get the log firing rate, then with the plus 1 noise on top that generates the spike. And the log firing rate is combined, depend on each of the inputs that are sort of external to the neuron. So this could be continuous inputs, so the position of the animal in the virtual reality, the distance to target, things like that. It could be discrete events like movement onset, movement stop, the delivery of the reward, things like that. And these have individual contributions to the firing rate that are non-linear. And we sum those up, and this gives us the log firing rate. Okay, so the model is not the most interesting part of this. It's how we do the estimation that really makes it powerful. So for the nonlinearities, we're going to use a spline basis. And with the fancy penalized log likelihood that enforces smoothness. So if components are not used in order to explain neural responses, that's going to flatten that nonlinear function to a zero line. And technically, it's actually non-trivial to get it to work exactly the right way, but we managed. In order to do that, we need to jointly optimize both parameters and hyperparameters using this, again, somewhat complicated estimation scheme. But at the end of the game, what we get is the series of parameters that we can use in order to build confidence bounds whether or not individual functions actually have a statistically significant contribution to the spike responses. So to show how this works in practice, here's a demo from some simulated data. And this data set has 8 inputs. These are temporal events. These are continuous time varying task variables. And we split them into subcomponent in the top that are actually driving neural responses with some nonlinear tuning function. And another set of responses that don't drive neural responses, but which are correlated with the first lot. So this is important because in the context of naturalistic behavior, like you don't factorize your input space anymore. So those are naturally going to be correlated. The distance to target, the speed of the animal, the acceleration, all those things will be tied together. So there's the danger of sort of leaking out dependencies in ways that you don't want for interpretability. Okay, so this is what the ground truth filters look like. This is what the estimator shows for the significant responses. And again, here you get confidence bound for each of the elements. Now, what happens with the non-significant ones? So again, you're going to get flat lines with a pretty tight confidence bounds. So if you integrate over these lines, you can compute the p-value that tells you what's the probability that this particular element, input element, is statistically significantly driving a neural response or not. And again, the p-values are going to be very small for the first 4, and they're going to be very large and not significant for the last ones. So the model correctly identifies that these extra 4 values are not actually doing anything. It's sort of, because it's a sort of a proper statistical test, it doesn't generate false positive beyond what you allow it to. With respect to how much data you have, it actually doesn't take that much data in this particular example to figure out the correct structure. If you have very little data, you might miss structure that's there. So you decide it's insignificant because you don't have enough data to constrain it. But generally, it does a pretty good job with relatively little data. I should say that we have 3-hour sessions long. So for monkey responses, we're way that way. And we try to downsample the monkey data and repeat the same government procedures, and we get very consistent kind of results. Where we use the 3-hour long data set as the ground truth and sort of 20-30 minutes long sessions. Yeah.
Sorry, I think I only missed a little bit. So what's the that's a filter here.
Yeah, so these are the nonlinear filters that get summed up in order to get the log firing.
So where is the nonlinearity here? Because it's a filter, so it's a convolution, I guess. Maybe all this.
So there's this kind of nonlinearity is the shape that's sort of generated by a spline basis. There's an additional sort of link function nonlinearity, which is the exponential that maps the log firing rates into actual firing rates for the possible nodes of the observation model.
But the filter is a convolution, right? So the operation of the filter is linear, or is it not?
But the shape of this filter is not linear.
Sure,
the operation is linear.
Yeah, and then while at it, I didn't really get the second line, so I see the first, they're flat.
Yeah, they're flat. Like
So-
we make them flat, so they're inputs into the model. They're correlated with variables that do actually affect the neural responses, but they themselves do not directly affect neural responses.
Yes, it's okay. I see.
Yeah.
So if they're perfectly correlated,
Sorry.
if
Yes.
you are perfectly correlated, then the model will be able to fit it correctly, right?
Yeah, so there's an explaining away effect that once it figures out that the true good variables are driving the responses, it doesn't need the other guys. So the regularizers punches them down to zero.
Okay,
It's kind of
so
the
you need to
defense.
assume that there's some correlation, not 100% correlation.
Yeah, if there was 100% correlation, then you were just putting the same input twice into the model, and that would be a bad thing. Don't do that. Yeah.
So in your formula, in the previous slide, the previous slide is the full formula.
Oh yeah, but you can go back to the.
Alright.
I'm getting some gymnastics as well. Like,
So here, the non-negatives in the F function, that's basically your spline basis here, or is that something?
yeah, so these are all approximately base practices.
I see.
So it ends up being formally a GLM, so it's not so much about the functional form of the model, but it's about the fancy estimation procedure that really gets the job done. And this is my excuse to move past this. Yeah, so the question is, okay, do I really need all this complexity, or could I get away with just putting a strong L1 regularization on the basis function elements? And the answer is that it doesn't really do the job. So again, on simulated data where we really know what the granular statistics are, like the L1 regularization still has a lot of noise in the estimation of this null dimensions. So it's hard to figure out that these things are really non-zero. There's also a small but quite systematic improvement in the ability of the PGAM, what we're calling our thing, model to fit the data in terms of pseudo squares. And this like persists even for really large data sets. So it's not that we're in a data limited regime where things are noisy to begin with. It's like even for relatively complex things, the estimation procedure seems to, or this regularization procedure seems to do better. Okay? The other thing that's sort of practically very useful when you're dealing with these very big data sets like the Firefly class is compute requirements. So fitting one of these models is a little bit slower than doing the GLM than one regularization. But remember that in order to use the GLM paradigm, as in the Hardcastle, you would have to fit an exponentially many number of models to do the mark comparison. So once you take that into account, then like that approach really scales miserably, and ours is much more stable as a function of both data size, so how long the recording session is, and also the input dimension, how many task variables are you referencing against. Okay, so it works. We're happy enough based on the simulated data. We have a lot more experiments of simulated data in the paper if you want to have a look. But what we really care about is, does this tell us something useful about what monkey is doing in the context of the Firefly task? And again, these results are available on the biography, so if I'm skimming some of the details and you want to know them, just have a look there. But this is how the data looks like. We have simultaneous recording from 3 gray regions. These are spikes from single units in the 3 regions. And sort of like the yellow is the one that the Firefly is visible. Then when the monkey stops in order to try to get toward the black dots, and then the spikes are in different colors corresponding to the different gray regions. There's a lot of variability in the duration of the trials. And again, there's a lot more variability about what the optic flow is, where the target is supposed to go, what the monkey is doing. So this varies along many directions. Okay, so why these 3 regions? So MSTD is a visual area, so it's supposed to be sensitive to the optic flow information that we're sending into the system. 7A, you can think about it as a motor area that's supposed to control what the monkey's doing. And the PFC, you can think about sort of this computing of cognitive variables, decision-making, things like that. So we're kind of trying to target regions that are historically known to be involved in aspects of this task.
So MSTD is also a multi-sensory responding to vestibular.
Yeah, there's no vestibular input in this version. like DORA has a way of getting the similar information to monkeys by having one of these like chairs that actually move when you think. It's very fun if you want to try it yourself. But in these experiments, that's not happening, so that there's not real similar feedback in this version. Okay, so this is the p-gap instantiated in the context of this data set. So if you have a model that can handle high-dimensional inputs and that can figure out itself from data which elements you need or not, the good strategy is just throw the kitchen sink at the problem and let the data like sort it out. So this is what we did here. So we're fitting p-gaps where the inputs are all sorts. So there's some monetary variables like velocity, acceleration, things like that, times when the movement is initiated, stopped, etc. The latent variables that are critical for the animal to compute in order to do the task, like the distance to target in distance or Euclidean angular components to that. But also, and sort of like the arrival of reward, eye tracking information. We're also throwing in internal information that we have as measurements at the same time. So this could include like filtered LFP in different bands from the same region or from different brain regions. Here's where the coupling or the pairwise correlations between neurons comes into play. We're also throwing into this model the spiking activity of all of the other neurons that we feel like it. Again, this includes simultaneously recorded neurons from the same region and also sometimes simultaneously recorded neurons from all of the other regions. So it seems a little bit insane. You end up with a model that has a few hundred input dimensions. But we were still confident that the estimator really works well in this regime. And I'm not going to show you how we know that, but I'm just going to do a little visual demo. So this is actual data, spiking activity of multiple neurons in a little chunk of time, a little bit smoothed. These are the predicted firing rates of the model that gets these inputs, not the other coupling inputs, only the task variable inputs at the same time. It's a pretty good match. So I think we're reasonably confident that we're good at capturing the key variability in these neural responses. Okay, so what do we get out of it? So first of all, we get a bunch of filters, some of which are non-significant, which are these very, very shady colors, and the more contrast high colors are the ones that are statistically significant, as in by our test. And what we find, these are example units from each of the areas, so it's not really conclusive, but based on this example alone, you can figure out that there's selectivity to everything everywhere. So mixed selectivity is really prominent in this data set, and what's particularly interesting is like if you're looking at this officially visual area that has pretty significant responses to cognitive variables. That's not supposed to happen according to the test. So we can quantify this in different ways. Here we're just looking at which fraction of the inputs are statistically significant. So not the shape of the filters, but whether or not they're significant or not. And this is statistics across all of the neurons that we're recording from. What you find is, first of all, MST looks a lot like PFC in many respects, which is kind of traumatizing. But we also find, again, selectivity to everything everywhere. What's particularly interesting from our purposes is this representation of the cognitive variables like travel distance and so forth, which is kind of in the 40–50% of the neurons are showing selectivity to these things, which is more that the highest number that we knew about before was coming from bat hippocampus, like running around trying to cover really large distances. And yeah, these numbers are bigger. So we're finding a really strong representation of these cognitive variables even in MST, which is kind of amazing or strange. The other thing that you can do, like you can treat these filters as a compact representation of this original huge spiking data set. So instead of having the spiking activity of all of the neurons, now we have a collection of little filters that correspond to each of the elements. And you can treat us like as feature vectors and embed those feature vectors in low-dimensional spaces so you can get a sense of how similar in the details is the tuning function of a neuron in 7a versus a neuron in DLPFC versus a neuron in MSTD. And here again, like this is what we have here, so it's a nonlinear embedding into dimensions. Each of the little dots correspond to one neuron, and the color corresponds to which area are coming from. So you get the sense that although there's mixed selectivity overall, it's not exactly mixed selectivity in the same way. So the 7a patterns of selectivity are different from the ones in the LPFC. And MSTD is doing weird things that have similarities with both, which is just weird. Okay, so this is what we have. Now, we can also look at the patterns of interneural connectivity. Here is the one based on LFP. So this asks, what's the probability that LFP in area A significantly drives responses in area B beyond what the local LFP would say? Okay. And we find some interesting patterns, and these are interesting because they respect to a large degree known anatomy about which area connects to which. So these guys are strongly connected to each other, these guys are strongly connected to each other. That seems to kind of be recapitulated in this kind of measure. Now, what's also interesting if we actually look at even at a finer detail at the patterns of neuron-to-neuron interactions. So here things are becoming a little bit like sparser, but you still have significant connections across areas. These are not going to be like neurosynaptic connections, but they're sort of like patterns of statistical dependencies across areas. And this also recapitulates what we know from previous work using more traditional methods, which is that the more similar the neurons are, the more likely it is to be for them to show this pattern of functional connectivity. Okay. The last thing that's interesting is that we didn't know about this before, and maybe there's something that we want to pursue experimentally more, is this strong pattern interconnectivity between MSTD and DLPFC. And the reason for that, why that's interesting is that anatomically there are no direct connections between these two areas. So this seems to reflect something about the dynamical system organization of the circuit in the context of this task. So that's something that we're trying to pursue further. Okay, so the last bit of interesting experimental evidence on this front is this manipulation of the sensory information. Sorry, I messed up my animation. So the monkey can still do the task, although there are going to be some changes in the biases that we can record. And if we look neurally, most of the time what we find is the overall shape of the tuning functions tends to be not that different, but there are systematic changes in gain. So things could go up or down as a function of the reduction of sensory information. And this is the summaries to the population. So what we see is like big negative changes in gain in the tuning function of MSTD neurons in the context of this manipulation, which means that the sensory neurons are becoming less sensitive to the sensory information. Okay, in a way. Whereas the other two areas have smaller gain effects that tend to be positive. If we look at the patterns of interconnectivity, there the direction is different. So the recurrent connections within MSTD get a little bit stronger, whereas the one in 7A and DLPFC stay relatively the same in magnitude. Okay, so this is the pattern that emerges from this data, that as the reliability of the sensory input decreases, the reliance of MSTD on this sensory information decreases itself, while compensated by stronger but similarly patterned recurrent interactions. Okay, and we can quantify this, like how the similarly patterned part of the statement, by measuring the similarity or the correlations of the coupling filters across conditions. So a high correlation coefficient means that the overall shape of the filter hasn't changed at all, although it could change in magnitude. Whereas a zero coupling means that it's completely reorganized, so the shape before and after that doesn't bear any resemblance to one another. And we also have like null models that tells us what's the best possible thing that could come out in terms of distribution of what the chances look like. So what you find is that MSTD tends to be quite similar, so it keeps the same pattern of interactions, whereas the DLPFC and the 7A are reorganizing internally their patterns of interaction in order to compensate for lack of sensory information and sort of like rely from the internal model. What's even more interesting is that if you correlate this measure to the session-by-session biases, changes in bias that we measure behaviorally, so whether or not the monkey seemingly compensated for the manipulation and behaved the same way, or it was doing slightly worse in response to the manipulation, then we see correlations between this stability and the change in bias. So in order for the monkey to optimally compensate for the lack of sensory information, you want MSTD to stay as similar as possible and the other two regions to reorganize as much as possible. This doesn't tell you about how exactly it does it, but it does give you sort of interesting hints about where you're supposed to be looking for the interesting signatures of the computation. Okay, which is what we're going to do in a minute. But as an internet summary, what we've learned so far. So we know from this sort of more complex setup that the whole is not the sum of these parts. So if you probe the brain in this regime where lots of things are going on, even what was traditionally considered a purely sensory region will start to show interesting cognitive signatures. The other thing that comes from the functional connectivity analysis, that there seems to be something interesting going on in the interactions between MSTD and DLPFC in the context of this task that was not known before and we need to investigate further. And lastly, there's a circuit organization when the sensory evidence is scarce that happens when the animal has to rely on, or reflects the fact that the animal has to rely more on the internal model. And the degree of this reorganization does couple with measured behavior in potentially interesting ways. Okay, so this is very descriptive. The natural next step is to ask, this is going to be very time. The natural next step is to ask, okay, so can we get at the underlying computation more directly? Can you analyze population responses and their interactions across areas in the context of this task? And we do this in the context of a very framework that we have a manuscript for submitted to neurons. And I'm going to rush, but the general idea, what we're trying to achieve here, again, echoes a lot of what we've heard about so far. So we're interested in finding non-dimensional neural manifolds that capture the underlying computation. This is an example of what this looks like in another task that we've been playing with. We also want to understand communication between areas, which, again, historically, you would do via things like communications by the subspaces that capture shared variability between area A and area B. Okay, the problem with this data set is that even after you do dimensionality reduction, where you're left with something like 30, 40, 50 dimensions. So it's very hard to visually try to assess what's going on in the neural manifold with simple, like dumb, unsupervised methods. So the hint about how to go about this is, okay, don't look for axes of variability overall, but look for axes of neural variability that are actually interpretable in some way in the context of like relevant task variables, which people historically again have done with the mixed DCA. But this is not quite doing the job for us, mostly because the vanilla implementation requires the trial repeats, and we don't have that for obvious reasons that we've been talking about exhaustively. So we don't, we can't use it out of the box. We also don't like the fact that it relies on Gaussian noise and you type Poisson variability to be more of a spike counts, because we're looking at relatively fine timescale changes in the primary responses. I didn't say this before, but we're using typically 6 millisecond timings because those are at the rate at which we're capturing our behavioral task variables. So we don't want to go very large, which you would need for the Gaussian. Now this is the general form of the graphical model. It's not very intuitive. We're going to go into concrete examples later, but the general idea is that you have population responses from multiple brain areas. You have some task variables that you're interested in sort of understanding in relation to this activity. You want to partition the variability of the neural responses in elements that are specific to the region, elements that are shared across regions, and any of these latent variables could be partitioned into sub-manifolds that have to do with something about the task that you're trying to explore. So again, this is complicated and it's not a very intuitive one. What's important to know with respect to the structure of the graphical model is that we're using Poisson noise, that we're smoothing the latent variables using Gaussian process priors. So this part of the model looks like a Poisson GPFA kind of model. The shared variability and the relationship to the task variable is modeled via a probabilistic version of CCA. And we're putting this all together and doing inferences in it. So again, I'm not going to talk about the machine learning aspects of this estimation. I want to show you how it does in somewhat interesting, not within data distribution type of scenarios. So the first version that I want to show is the one where you have a single population, and in this population there's a low-dimensional manifold that carries task-relevant information. So the way we generate the simulated data for this is using latent dynamical system models with again, Poisson observations on top. And these are driven, some of these are private, some of them are driven by the external task variable. Okay, and the version of the estimator, which we somewhat lamely call TameGP, task-aligned manifold estimation Gaussian process priors, the model, is like this. So we're trying to partition variability in these spiking responses into stuff that doesn't have to do with the task and stuff that has to do with the task. Okay, so here's a big comparison of how this looks. This is the raw trajectory, the ground trajectory. This is what our estimator spits out. This is what other methods that we would use for similar purposes would look like. What's interesting to note is, first of all, with respect to reconstruction of the underlying data structure, our model does better than the alternatives. It also does better or similarly to PGPFA when it comes to actually capturing. It's really cool. How much yeah, I'm going to pretend that I didn't see that. Okay, so capturing the actual neural responses. You can do this also with shared activity between areas, where here again you have latent dynamical systems that communicate linearly between one another. And it still works better by different measures. What I think is practically really interesting is that when you're doing model comparison and trying to figure out what's the actual dimensionality of the shared manifold that exchanges information between these two brain regions, our estimator tends to be pretty much on the money most of the time, whereas other approaches, like the semantic approach, which is low-rank regression, tends to overestimate the shared variability, the dimensionality of the shared variability between data sets. And very lastly, we also do this on neural data, and we get more interpretable latent dynamics, better fits in terms of pseudo-R-square of the actual data. And this is the very last thing that I'm going to show, and then I'm going to end up. And we're trying to figure out, if you didn't have this tool, and you just did unsupervised latent dimensionality reduction, how much more dimensions would you need in order to capture the same amount of information about the underlying task variable of interest? So here we're looking specifically at distance traveled, which is either angular and actual, it's polar coordinates, so it's a two-dimensional vector. 10GP needs 2 dimensions, which is the same number of dimensions as the actual class of variables in order to capture that information. And this is decoding R², which asks, we do the dimensionality reduction unsupervised without task knowledge, and we ask here, how many, if I tried to get the same amount of information about this task variable from GPFA projections, how much better or worse would I do? So this captures a lot of information about distance traveled in the first 2 dimensions. This guy needs something like 10 dimensions to get even close. So this means that this gives you much more compact representations that are possibly visualizable or interpretable. This is very early days, so we're doing a lot more work on this. I'm going to skip this so you can ask any questions. But what I want to convey is that making sense of neural responses through complex behavior is really worthwhile, but a really hard statistical challenge. This PGAM and TMGP are our attempts to bring us close to a solution in this domain. And although they're really helpful in visualizing the data, getting some hypothesis going with respect to this data set, they're in no way the end of the story. Like, it pushes us to try to understand brain computation as nodes of dynamical systems interacting with one another. I think that's the right formalism in order to really target the computations themselves. And we don't have the tools to do that in the context of this kind of behavior, but this is what we need to get working to really get to the answer properly. And with that, I'm going to thank you for your patience and thank all these wonderful people for supporting our work, having sources, etc. Thank you.
Thank you, Cristina. Do you have any questions?
Yes.
Yes?
Yeah, really amazing. This shared selectivity, what is that? Do you know? It's really boring. It gets worse and worse, right? Every paper you see on this topic, it seems like that everywhere in the brain you can find anything.
Yes,
no, I mean,
you can be depressed about it or happy about it. There's more stuff to understand.
The question, I suppose, is did you check for those different? You just have a large collection of task variables, right? And do you know if are there any hidden correlations that you actually? I mean, these are not independent.
Yeah,
task
this
dimensions.
is why we did like a lot of the simulated work where we were trying to replicate as much as possible the structure. So in this particular kind of jobs, I think 90% of the estimation exercise is getting simulated data that matches as many of the statistics of the data set of interest that you can, and like checking the hell out of the estimator in that statistical regime to make sure that it behaves sensibly. Like you're never 100% sure that there might be some like a key statistic that we didn't explicitly check for that's really important and that affects things, but yeah, we tried really hard.
Yeah, like did you? I mean, what would happen in the case if you had 2 task variables that was correlated in some way? I mean, there could be something like a visual one and then some that relates to movement, right? So a simple regression would tell you they're both related. So you think, oh, there's movement in the visual system, right? If vision and movement are indeed somehow correlated. But in your model, is there machinery to try to sort of
so that's exactly what the first set of simulated results are. So if you have 2 correlated variables, one drives neural responses, the other one absolutely doesn't, it's going to find only one. Because of explaining.
Okay, good.
Like if they're correlated to different degrees, hopefully it will figure that one out. So yeah, we think that's a thing, but yeah, it is very tricky. There's one experiment that we really wanted to do when we had technical difficulties, is to take all of the tasks, like the internal model, all the computation out of the system, just to like simulate exactly the same sensory experience with the animal completely passive, not doing the task, on a like trial by trial basis match, and see how that affects the statistics. Like, presumably, if this is an active process that reflects the computation that the monkey is trying to do, it will go away in that regime. And I really, really wanted that data, but we have technical issues, and it's still not there. But yeah, I don't know. If you have other ideas about what we can check, I'm always happy to tell Eduardo to do it.
Any other questions?
Right, so in your model, you mentioned that you put in the behavioral variables and other neurons. So if you record enough other neurons, almost by definition, you won't need the behavioral variables.
That is true. I mean, like, presumably, the direct behavioral variable, if that's the one that thing really driving the responses, will still be a better explanator for the activity than the other neurons. So the explaining away phenomenon will still apply.
But functionally, maybe some of these neurons. So
yeah, so like this is sort of a fundamental limitation of a neural-centric kind of discussion. This is really a network phenomenon happening in the entire circuit. And this is why we need to move to population-level analysis to really understand what the function of these statistical observations is. So for us, we really treat this, the way experimentalists treat tuning functions these days, I think, as a hypothesis driving, like, trying to figure out, okay, well, like, if you haven't seen this kind of data before in the context of this kind of task, the first thing you do is you look at, okay, what do individual neurons care about to get the sense of what the hypothesis space is, and then you go through sort of more meaningful, more hypothesis-driven kind of analyses. But yeah, so you need to like set this in the context of this kind of complex task, you need to set these basic building blocks first, because the traditional way of doing it just doesn't work. So we're way far from the like solution of how the computation works, but hopefully these are helpful steps to help us along the way.
Right. So
Sorry.
no worries. At this point, we should wrap up our first day of the workshop. And next thing on our plan is to have a poster session. So probably we will have a short break to let poster presenters set up their posters, and then we can discuss them. And at 7, we'll have dinner at 56 North Restaurant.
Yeah, so perhaps we can meet here at 5 to 7 or so, and then go to the place. It was just in 2 minutes from here.
It's really just across the road. Thank you very much, Cristina.